Hey everyone. Bone health declines during aging. And in terms of why, most people aren't thinking about vascular health or the lymphatic system, but both systems impact bone health, regeneration, and aging. To learn more about those mechanisms, today's guest is Dr. Anjali Kusumbe, who is an associate professor of stem cell biology and regenerative medicine. Welcome, Anjali, and let's get into the presentation.
So, hi, Mike. Thank you so much for this invitation, and it's a pleasure to be here and to speak about what my lab is doing. So what we are interested in is we look at the vascular diversity and how the vascular diversity changed during the tissue aging and tissue regeneration, with the ultimate goal to identify therapeutic targets to treat the age associated diseases. And on those lines, I will be explaining a bit more about it and giving you two of the exemplar projects ongoing in the lab. So basically, we know that the blood vessels and the lymphatic vessels are important. They form the transport system. For example, the blood vessels play an important role in the delivery of oxygen and nutrients to the tissues. And but more importantly, in addition to be just a transport network or transport tube, they also play a critical role in the organ development, tissue repair and regeneration. And we also know that the lymphatic vessels are crucial for the resolution of the inflammation. So they are just not the tubes, they have more functions to them. And this is actually because these lymphatic vessels or the blood vessels have these endothelial cells and the pericytes, and these cells can engage into the justacrine and paracrine signaling with their neighboring cells. And these neighboring cell in a tissue could be a stem cell, it could be an immune cell, and if we talk about a tumor context, it can be a cancer cells. So by engaging into the paracrine or justacrine signaling with the neighboring cell type in the tissue, the vasculature can, or the endothelial cells can regulate the fate and behavior of the tissue and thereby the function of that tissue. And these are the kind of interactions my lab is looking at, and particularly what we are also interested in, how with age our vasculature change. Is the lymphatic vessels changing, how the blood vessels are changing, and we are investigating it, the changes in the vasculature. And today specifically I will be talking about the lymphatic vessels in bone and the aging of the skull and the skull bone marrow, and how the blood vessels play a role there. But before even starting with these areas of the projects, what I wanted to talk about is what is known about the vasculature in aging so far. So it has been quoted long back by a British surgeon, Thomas Sinthiam, that we are as old as our arteries. And by this, he meant that as we age, our arteries stiffen. But until recently, not much has been known how the lymphatic vessels age or how the capillaries, the microvessels age. And this has been a hot topic in in the vascular biology field to in the last 5 years. And we have been also investigating this. And today, for I will take bone as a tissue as an example to study the aging and the regeneration. Why bone? Because bone hosts these blood stem cells, and we know that these blood stem cells can give rise to all the blood and the immune cells in the bone. It also hosts the skeletal stem cells, which give rise to chondrocytes, osteoblasts, and adipocytes. And for both of these stem cells, it has been shown that these capillaries, the microvessels, form a niche. They form a support for the blood stem cells and the skeletal stem cells. And this is also the case of the many other stem cells in our body, that the vasculature is forming a niche and the support and supporting the stem cells. The other reason I'm interested, my lab is interested in bone, because bone has a higher capacity to regenerate, to remodel as compared to the other tissues. If we say brain, or if we say heart, we know that the bone can regenerate, our fractures heal, but We also know that as we age, our capacity to heal the fractures decline. So elderly patients have problem with healing the fracture. There are non-reunion fractures and bone marrow transplantation. The other aspect is also a problem in the elderly. So even a highly regenerative tissue like bone is declining its regenerative capacity with age. And this is why bone is a good tissue to study tissue regeneration and aging. And my lab have we follow this approach of whole bone imaging and tissue clearing. The reason why is that when we talk about the vasculature, we and the interaction with the surrounding cells, we are interested in the spatial analysis. And this is for this it is very important to have the 3D context. To have this 3D context, we do the tissue clearing, we do the light sheet imaging to understand how the vasculature in 3D is interacting with the stem cells. But when we talk about bone, imaging has always been very difficult, and you can imagine this because bone is hard compared to the soft tissue. So first it needs to be decalcified, made soft. But then the decalcification process comes with a huge limitation because many of the decalcification protocols are very harsh. Then it affects the antibody binding. So there is a limitation here, and that's why the antibody binding and the immunostainings becomes challenging. So many of the protocols which have been people have been using depends on the use of a reporter mice and so on, so they don't have to do the immunostaining. But then it is very limited. One can only analyze specific markers, and the imaging in the bone in 3D and in the whole tissue context becomes very limited. And to overcome this challenge, we developed, I will not be going into much of the details of the protocol, but we have developed this ultrafast imaging method for the intact bones and all the skeletal tissues like joints, where we can very quickly in 3.5 days clear the bones. But not only we are clearing and making it transparent, as you can see this murine bone has now become transparent, but we can also do the immunolabeling. So we are not depending now on the reporter mice. We decalcify with the softer, like less harsh method. We are doing the imaging, and we can not only do for the mouse the murine bone. So here is the murine. So this was the murine bone. This is the murine long bone tibia, and we can see with the red is the staining for all the capillaries, the green is the staining for the arteries. Since my lab is interested in blood vessels and the lymphatic vessels, we are looking at that. But one can actually use any antibody and look at the populations they are interested in. We can also do other bones listed here, but most importantly, this method is also useful for doing the human bone biopsies, and we can image the bone now at a higher resolution. So the old paradigm, when we talk about the skeleton or we talk about the bone, is that there are no lymphatic vessels in the bone. And since we were looking at many markers and we were doing imaging with our newly developed method, we also looked at one of the marker, and we were surprised at it when we looked at a marker called as lymphatic vessel endothelial hyaluron receptor 1, which is LIVE1. We saw that it is being expressed. It is present within the bone. Like this is a murine long bone tibia. But more importantly, these are all the tubular. You can see that they are the vascular structures we presented, and we were quite surprised. And we looked into different contexts that they are not only on the surface of the bone, they are penetrating, they are inside the bone, and this is the cross section of the bone. And we can see that all these green staining is for the LIVE1. We can see these LIVE1 positive vessels are within the bone. If you cut the bone like this as a cross section, we can see that they are penetrating the bone, they are inside the bone. And since the LIVE1 marks also the some of the macrophages, we have also excluded the presence of macrophages by staining with the macrophage markers. So basically, these Live-1 positive vascular structures are definitely lymphatic vessels. They are penetrating inside the bone. And if we look at, and interestingly, if we take the public databases where people have done the single cell sequencing of the bone, we can see that indeed in these public, so this is not generated from my lab. So we took some of the public databases, and this is just one of the exemplar database. We see that the lymphatic endothelial cells are present in the bone. They express all the canonical markers of the lymphatic vessels, like they are FLT4 positive, they are PROX1 positive, and in addition to Live-1, the PROX1 is very important because PROX1 is the transcription factor. It is Prospero homeobox 1. It's the transcription factor which is required for the lymphatic vessel identity. So if the endothelial cells express PROX1, they will differentiate into the lymphatic side. So we can see that there are lymphatic vessels in the bone. Their existing databases shows that. And as I said, we can look at these in the multiple database which are existing. There are PROX1 positive cells there. We also found that when there is a situation when the bone is regenerating, for example, here I am showing an example of injury and the genotoxic stress. Specifically here, this is the mouse treated with the radiation, the lung bones. If you can see, in green are the lymphatic vessels. After the radiation injury, when the bones are regenerating, there is a rapid expansion of the lymphatic vessels in the bone. And we can also see that even if we take the thin tissue sections where it is difficult to look at the lymphatic vessels, because in the healthy bones they are sparse. But if after the injury, so it is again a public data set which is available where people have done the spatial imaging on the thin sections of the mouse bone after the fracture. We can again see these PROX1 positive cells that they are there after the fracture, even in a thin section of 5 micrometers. So they are basically present, and they are the endothelial cells expressing other endothelial markers. And this is the other data sets where we see after the fracture injury, after the fracture, there is a presence of the PROX1 positive cells, which also shows now, see, since they are visible on these thin sections, they are expanding after the fracture injury, because it's very difficult to see them on the thin sections of the bone in the healthy bone where they are sparse. But again, I will not go into the much details of it because this work is published, it's available. But what I would summarize here, that we have discovered that lymphatic vessels are present in the bone. They are not only present there in the bone, but they play an important role during the injury and the stress situation. So during the injury or the stress, for example, the fracture or the radiation stress, they would proliferate, the lymphatic endothelial cells would proliferate, and then they secrete the niche and the supporting factors like CXCL12, which then is required for the hematopoietic stem cells for the hematopoietic regeneration. But also they are required by certain pericytes, which then differentiate into the bone cells. Again, in the interest of the time, I will not talk about much on the aging for this project, but what we have also found that the lymphatic endothelial cells in the aging mice, in the aged mice, even after an injury, they are not proliferating because the cells are more senescent. They lose their capacity to be activated or triggered by the stress and injury, and there is no proliferation after the stress and injury. And that's why we also see that the old bones have a delayed capacity or lower capacity to regenerate. We also see the lymphatic vessels are not proliferating after the stress and injury. So now the next exemplar project, which I will, because I've been talking about the lymphatic vessels so far, I will be talking more about now the vasculature. And this is a project which is not yet published, but hopefully will come out soon, but is not yet available. So what we are looking at here, how the vasculature and the microcapillaries and their role in different bones differ across aging? So is the capillaries and the microvasculature similar across all our different bones? For example, a flat bone skull versus a long bone femur and tibia. So this is one of the project which I will be discussing. And we got specifically interested in the skull and the skull bone and the skull bone marrow. And now, why skull and skull bone marrow? Because it has been recently proposed that the adult skull bone marrow might be an expanding reservoir and resilient to aging. So, which means that as we age, the skull is forming the hematopoietic reservoir, and the main conclusion of this study is that the vasculature expand with age on aging. So there is an expansion of the vasculature, and since there is an expansion of the vasculature, it's resilient to the aging because the vasculature is forming the niche for the hematopoietic reservoir. But since we were investigating other different bones, when this paper came, we were very interested in this because it has been also reported by various labs that as we age, the skull loses its capacity to repair, to regenerate. There is senescence, and there is inflammation. So it was kind of a mystery that how is the vasculature expanding and what is so special? Why only within the skull the vasculature is expanding? So we then, because this has been something, we actually investigated this with various groups, and what we found here is that if we look at this paper, which has as I have previously mentioned, they have shown that as we age, see if you see this endomucin and CD31, there is an expansion of the vessels. So CD31, endomucin are all the vascular markers. So basically, the vasculature is expanding upon aging, and there is something special in skull and this is why the vasculature expand, but because every other bone there is no expansion. So what we did, but then we also saw that this work dependent on the whole mount imaging of the skull without applying any tissue clearing. And I have previously shown you that tissue clearing and making the bone transparent or any tissue transparent is very important to image the whole structure throughout. Otherwise, it's not possible to image the structures throughout without if it is transparent, and particularly it's impossible to do this with the confocal microscopy because confocal microscopy is limited by the imaging depth unlike the light sheet microscopy. So we then investigated with our methods more into in the skull bone, and today I will be showing you some of the regions of the skull bone, which is the parietal bone. This is parietal bone in the calvarium and the intraparietal bone. So what we again did is looking at the imaging, and what we again see here, if we look at the imaging, what we found is that if these kind of methods are leading to the artifacts, which means that where I have previously shown that they have done this immunostaining with the endomucin. So endomucin is a cell surface marker. It's a well-known marker of the capillaries, and it's also only present of the skull. And if the methods, because are not good enough, especially with the bone where it's a difficult tissue to image, for example, here it's seen that, and this is from the same paper, the endomucin is accumulating in the nucleus, which is not biologically possible because it's not a transcription factor, it is a cell surface marker. And if we do a immunostaining for the endomucin, we can see the endomucin immunofluorescence is restricted to the cell surface, which is expected, which even within the skull and the calvarium. So there are imaging discrepancies, and then we investigated based on this further with what is happening between in the skull, the different regions of the bones, not only by the light sheets imaging, but more importantly by the tissue sectioning and imaging the tissues at the different layers, so that we can see from the first section to the last section. So here, instead of the whole tissue, what we have taken the approach is looking at the consecutive sections from the first to the last of the skull and see what are the changes in the vasculature. And based on this, we do not see any significant expansion of the vasculature with age, which is also coming out in the same thing. If we look at the data sets which are within the paper, we see the same, that within the paper published as well, we see that the vasculature, if this in green are the endothelial cells, in fact, the single cell sequencing data sets do show that with aging, the vasculature declines. So if you see this, there is a huge decline of the vasculature. There is no increase. So we see that the skull in no way is special. There is no increase as we see by imaging of the capillaries. But more importantly, even in their single cell sequencing data sets, we see that there is a decline of the endothelial cells with the aging, which is also in line with the declining of the mesenchymal stem cells and the osteoprogenitors with the aging. So why I'm saying mesenchymal stem cells and osteoprogenitors, because they are also the important niche cells, and these stem cell and the progenitors are also declined. So skull, we do not see in any ways is protected against age, protected or resilient to aging. And we can also see the same thing if we look at the different, if we score and do the unsupervised analysis, we see that there is upregulation of cellular senescence. OS is the old skull. We see there is an upregulation of the cellular senescence. There is negative regulation of the telomere maintenance, and there is epigenetic regulation of the gene expression increases in the old skull. And again, I will not go into the details of this, but in the interest of the time. But if we also do the if we also do the unbiased proteomics analysis, we see the same. We see where that there is a increased DNA damage in aging. There is an increase in the inflammatory response in aging. So this is the proteomics analysis of the skull, and we do see that the inflammation, DNA damage is increasing, osteogenesis is decreasing. Similarly, angiogenesis, which is the vascular growth, is decreasing, which is all fitting all the data with the imaging, their own single cell sequencing data, and this proteomics all is combined, saying that there is nothing special happening in the skull. It's not resilient to aging, and the mitochondrial activity is decreasing, because all this red is for the upregulation and the blue is for the downregulation. So DNA damage, inflammatory response is all increasing with aging. So so far, what I have shown you, that with aging, there is a degeneration of the skull bone marrow niche. There is DNA damage, senescence, inflammation, there is adipogenesis. So we do not see skull being able to expand vasculature or expand angiogenesis and being resilient to aging in any of the context when we look at the hallmarks of aging. But if we look at different bones in the mouse, what came up, again, I am using the word is the relatively protected. So if we compare different bones, OV is the old vertebra, and if we compare old skull with old vertebra, we see that the DNA damage, mitochondria, the DNA damage and the inflammatory response is basically lesser in the vertebra. OV, which is the vertebra, is lesser in vertebra compared to the skull bone. And here we also see the mitochondrial activity is higher in the vertebral, old vertebra. So basically we see that the skull compared to skull and compared to vertebra, there is, if we compare, they are relatively protected. So vertebra, and I am not showing you the data of the long bone, But we have also compared skull, vertebra, and long bone, and we see the same. Out of all the three, the vertebral bone, the vertebra is relatively protected from the hallmarks of aging if we compare. But still, if we compare young versus old, there is an increase. But if it's just it, one has to be mindful that I'm only saying between the old bones of the mouse, not comparing here the young and old, where we see that even in the vertebra there is more senescence and all these are increasing. So what we have actually, so what I have shown you so far with the first part of this project is the aging. There is a decline in the skull angiogenesis, vascular expansion, but also there is degeneration of the skull bone marrow niche. And if we compare with skull and vertebra, we see that relatively there is some protection in vertebra for the hallmarks of aging compared to the skull. With this, I would like to end my talk, and I am open for the question, and I am hoping this work will be out soon.
So that was great. Your star, I think your lab is going to produce even more great work. So I was excited to see that presentation. So I think I'd like to start with and some of these may not have answers, right? So practical applications. So, weight-bearing exercise can help, sustain bone mass. But does that then lead to a better stem cell niche in delayed bone aging? Or is this just no matter how much you exercise, it's only a matter of time before the underlying mechanisms, whether it's a loss of the vasculature or a loss of the lymphatic system in bone, that it's only a matter of time before bone degenerates because of other mechanisms. So is exercise central to this or not?
Yes. So it has been shown that exercise increases blood flow, promotes angiogenesis, and angiogenesis is coupled with osteogenesis, which is bone formation. So if there is more exercise, there is if more angiogenesis, and exercise does help in keeping the bone healthy and keeping the bone mass, maintaining the bone mass to a certain extent.
But the challenge there is the skull bone, right? Because weight-bearing exercise, like, if I do some push-ups, I'm getting my arm.
Oh, you are
Yeah.
talking about the skull bone. But
Yeah.
then, see, the skull bone also has this advantage because the skull bone is also like the long bone is always under mechanical stress. And if you would have heard about it, vertebra is most affected by osteoporosis, not the skull bone. So they have different kind of osteogenesis, and the skull bone is not that much affected during aging first because it has less, at the same time it has less mechanical stress. So I think so it's being in that context it's less affected by osteoporosis. Vertebral bone is more affected by osteoporosis because it's this bone where the weight bearing it's a bone.
Well, so in mice, it makes sense because the skull bone, the vertebra, they're
It's
parallel
not.
to the
Yeah.
floor.
Yes.
Whereas
Yes. Yeah.
we
Yeah.
can do
Yeah.
weight-bearing exercise and put
Yeah.
stress on the vertebra to help,
Yeah.
build vertebral bone density. But the skull bone is not, I mean, nobody's really standing on their head. I don't even know if that
Yes.
would increase bone density. So
Yeah.
wouldn't that suggest that the skull bone is somewhat protected from aging in terms of a loss of bone mass?
Is it protected? You are saying we don't see it's protected.
Or less a lower no, right, I get that. uh I think you said that the skull bone there was less loss of bone mass relative to vertebrae.
I mean, in general, if we see osteoporosis, I'm not saying based on our study, I'm saying like osteoporosis, if it happens, it is which bone is affected most is our backbone, this bone, isn't
Hmm.
it? So in that context, I'm saying it's not for the hematopoietic niche. So like here, what we are talking about the niche for the hematopoietic stem cells. Where are the hematopoietic stem cells? So in context of osteoporosis, it's different where the bone mass and bone density and the strong bones are important compared to the hematopoiesis, where the niche is important, the supporting niche to promote the, so which could be different, where, for example, vertebral bone, it is known to have the red bone marrow, it's known to have all these hematopoietic cells, and it's, so it might be a site, it is likely to be a site where majority of the hematopoietic stem cells are residing, rather than in the skull, because skull is forming only a secondary compartment for the hematopoietic cells, the hematopoiesis, where all the blood cells are formed, and all our red marrow and all, is in the vertebra.
So is the red marrow red because of mitochondrial density or some other reason?
I think it involves a lot of things, how the blood is produced, but also it involves the context such as adipogenesis and so on, how much adipogenesis and the fat is deposited. So it's more, it's like the fat deposition and so on.
So fat deposition will control how yellow it is. So
Yeah,
if there's less adipogenesis, would the more red suggest greater
yeah.
mitochondrial density? And would that be the prime factor for it being?
So less adipogenesis means more of the hematopoiesis. So more of the hematopoietic progenitors and the stem cells, more of the red blood cells production and so on.
So
Erythroids.
would the hematopoietic stem cell niche with the adipocytes and all the other cells that are there, stem cells, blood cells, or that can become blood cells, is that because when I hear red bone marrow, I think mitochondrial density, even though that might not be the major story there. So I wonder if the different bone repositories, whether it's skull versus vertebra versus like femur, have I think you showed that a skull versus whichever bones you're looking at, it's still going to degenerate with age. But is that true across the board? And are there some bones, whether it's vertebra or others, that are more resistant? And would that red bone marrow, whether it's mitochondrial density or whatever the mechanism is, be a part of that story? Like, can we learn something from bones that may be more resistant to aging and degeneration by the niche that is enriching them?
Yeah, so basically, for example, if we look at, if we compare skull, vertebra, and long bones, we see the vertebra is relatively protected. But again, we do not see in vertebra that there is an expansion of blood vessels. So we are still looking for the mechanism. Why? Because it's definitely just not the number of endothelial cells or number of capillaries. Because we do not see expansion of the capillaries in any bones with aging, neither in skull, but neither in not also in vertebra. So that is definitely not the thing. So there is something else which is different in the vertebra, which is not definitely the endothelial cell numbers and the capillaries, which is there where the vertebra is forming at least the reservoir for the hematopoietic cells, and it's a major site of hematopoiesis in even in aging
So
now.
when considering that you showed data for mitochondrial dysfunction in the niche, have you looked at isolated mitochondrial function relative?
Oh, not yet. So we are just doing at the tissue level. We have to again. This is, as I said, this is very new. This is not. I mean, this is something we have to look into it. And I do always get the questions for looking at the mechanism, which is always difficult to investigate in this context, because again, it's new. But we are trying to understand why we see that different aging trajectories, because we can definitely say that with all this profile, which was also not known, that's all these bones are actually having different trajectories of aging. They age at a different rates, they are not aging. So that is, I think, so main take home message from here is that what we learn, which is very new, that even if there is a report that skull is protected of aging resilient, but it's not the case. It's not just protected or something. What I think, so what we are learning from all this is all different bones are aging at different rates, different trajectories they have. So some bone may be good for osteogenesis and all, some are forming hematosis. I think so they might have a specific function or a specific cell types they are contributing. Like if there is a myocardial infarction, it's likely that the sternum will form the site to provide the immune cells or the neutrophils at that point rather than coming from the long bone. So, because it's close. And similarly, if there is a stroke or something, the skull will provide a site to give some immune cell, which already people are studying and sure. So, but then it's not, the skull is nothing special about it. It's sternum can, we have to look into it. It's likely that the sternum can do for mito. It's like, so we can't, because, we are, unless we study it in the whole context, we are not, like I am now saying, that's why I'm making it very clear. I'm saying that skull, vertebra, and long bone. So it could be that on the next year we study more bones and we found that sternum is even protected. So I think so. It's too new to make that conclusion that the vertebra is best. But yes, if we compare long bones and skull and vertebra, it's correlatively. But what we most importantly what we learn through this project is all bones are aging at different trajectories. So when I talk about skull, all our data is focused on calvarium, and I think so. Mandible is also very different. This bone is very, we are doing all these studies and we see that the mandible is very different. So I can't even generalize as skull. But I use the term skull because in literature, a lot of people have used this. Even this paper which has come, the title itself says the skull, but that is not correct because the calvarium is very different, which we have done for some of the initial assays from the mandible. So and it would definitely, the calvarium will age in a different rate to the mandible. So I think so the main take home message from all this is, which we can be very sure of, which is also very new, which people did not know, that bones are aging at different trajectories, have different aging trajectories, and they are compartmentalized. Yeah, so everyone can keep their different aging states. That is also new, isn't it? There's less there is a compartmentalization. And so the third thing is that it might age differently for different things also, like different cellular aspects, whether it's a niche versus the hematopoiesis versus the osteogenesis and so on. So I think so that is, and then it is much more complex to understand with the mechanism, because I think so it would require, as I'm saying, comparison with more bones like sternum, here all studies done with calvarium, but we have to compare the mandible, for example.
The mandible versus the top of the skull is an interesting point. I mean, when you think about chewing, that could
Yes,
potentially
yes,
build,
it can.
couldn't that build bone density? I mean, you're gonna,
Yes,
you know,
and then if you see the density is higher compared to here, so that would be something. Yeah, it has this chewing activity, isn't it? Yeah.
and then so mice, for example, I think the data is similar in people where people chew less, and then whether it's taste or whatever, it's just they're less able to chew, and then that's going to contribute to the further decline of bone density here. But the bone density here is an interesting one. This goes back to my original point of how do you build bone density in an area that is not weight bearing, the top of the skull, which suggests that maybe it's completely dependent on the niche. And if the niche and the stem cell niche and lymphatics and vasculature goes away, well, you can see, you look at older people and the bones in the face have thinned, so is this
Yes,
a,
so that is cosmetically like the aging signs on the face is because of the bone loss, isn't it? In fact, there's some people even ask me, can you retain the bone, and then the face doesn't, the how the sagginess of the face and all those comes because the bones are, so that is other thing. So there is for sure, there is a, even every bone is losing bone mass with aging. So even our face bone, every bone even is losing it's not only the long bones or the vertebra, but then there is how they lose is differently, and how, what is the impact is also different, isn't it? When our long bones are affected, or the backbone is affected, it has health implications compared to where the face bones are affected. It's just the implications on looks, isn't it? The
Yes,
age.
cosmetic.
Yeah.
Yeah. I mean, I don't want to say fixing, but optimizing bone density in the legs and spine is easy. I mean, you can just exercise, but that's, the top of the skull. But as you mentioned, cosmetic. So I guess then regeneration comes into play and, regenerating the niche, whether it's the endothelial cells, lymphatic endothelial cells. Are there plans you have? Maybe do you have like a years forward plan to look at ways to regenerate the niche as a means for bone regeneration, whether it's in the long bones or the vertebra?
Yeah, I think so. That is where we are trying to look at whether we can promote specifically angiogenesis in certain regions of the bone, because we do not want to do it sometimes glow, like, uh, we need to promote angiogenesis only in the bone. We don't want to do it whole systemic, like, systemic angiogenesis increase in the aging. So that would require more bone specific markers, bone specific targets for increasing the angiogenesis, and that is what I mentioned in the first slide, that we are looking for the such targets, the molecular targets which we can use and exploit for during aging. So this would require understanding more about how we can have a target or some molecular targets which we can exploit only for the bone. We do not want to increase angiogenesis and vessels all over the body.
So would
And
that be
also, yeah, so that is something ongoing. We always try to understand why the vasculature in bone and how the vasculature in bone is different from the other organs, and how we can specifically target them.
So would that be like injections where your VEGF or some other angiogenesis promoting protein? Would you just inject that into bone?
Yeah,
You do that.
no, I don't think so. I mean, that is not a good solution to it, because VEGF, it has like, I mean, how many times you can inject, and who wants to inject the things in the bones?
How would you do?
So,
How would you do? Yeah.
yeah, so I think so. We need bone specific targets. We do not want something which is so systemic, like something which is global, glo- that it affects every blood vessels in every organ. So we want organ specific, tissue specific, bone specific things.
How would you do? How would you do that? Would you use the
No,
bone
no.
cell surface?
So we are not yet to that point, because we have to first, for that, understand at the molecular level what is so different about the bone vasculature, and what we can exploit without affecting. We know how the bone vasculature is different, but many of the molecules still are similar between the other vascular types. So we have to be having certain targets which are specific for the bone vasculature. So we do not have to inject in the bone. So even if it is taken, it only the vasculature in the bone will be responding.
So along those lines, or the niche, right? Could it also be the niche?
Yeah,
Not
yeah. So vasculature is a niche. So I mean, when I say niche, vasculature is a part of different cells can form a niche, and vasculature is one of the cell type which can form a niche.
So the one that comes to mind is atop the list is the shift of pericytes into fibroblasts. So would injecting more pericytes or
Hmm.
what's the mechanism? So I looked into this and, so I'm a big proponent of, so systemic microbial burden increases during aging. Older adults are like 10-fold higher incidence of bloodstream infections with bacteria, fungi, virus relative to younger people hospitalized with bloodstream infections. So I think that, and immunosenescence, now the immune system can't combat microbial invaders. So I wonder if that pericyte to fibroblast transition, when considering that fibroblasts have a greater antimicrobial capacity relative to pericytes, I wonder if that's part of the story where some microbes, whatever it may be, bacteria, fungi, virus, all of the above, get into the niche. When I say niche, in the whether it's in the vasculature or the lymphatics, and then the pericytes, differentiate into the fibroblasts in order to help fight that off. But the consequence is you need those pericytes as a part of the youthful niche to have good bone density. But now because you have the fibroblasts fighting off the microbes, now you have fibrosis and all of the other negative effects that come with that. So what do you think? What do you think?
Yeah, that's a very interesting hypothesis, and it could be, but also another way could be that there, since with aging the endothelial cells are senescent, are becoming senescent, they are not able to support the pericytes anymore, and then the fire, they because of those interactions, the the pericytes are differentiating into fibroblasts. And other thing which I also want to mention about the fibroblast and aging, and when you say this infection and so on, is that we are interested in understanding how the aged fibroblasts are different from the young fibroblast, but also those in arthritis or those in calves, like the cancer associated fibroblasts. Because I mean, I think so. It could, like, fibroblast is all about what are they, isn't it? Some fibroblasts are good, they are good in certain conditions, but like in arthritis, they are not good, they are inflammatory or so. So I think so. It might be also with aging, what are the kind of fibroblast, which are there good fibroblast or are there bad fibroblast, and so on, could also change the situation.
So are there plans to look at fibroblasts and whether it's
Yes,
metabolites
yes,
or
yes,
so?
yes. So that is an ongoing thing where we are trying to understand the signatures of the aging fibroblast and how it differs across tissues, but also how it is different from arthritis or other conditions. So they can't be same as like what are they in the arthritis, for example, or fibrosis.
Yeah. So do fibroblasts produce cathelicidin or antimicrobial peptides that you could look at in the arthritis or other, aging related
No, no, no.
conditions?
We are not looking into arthritis. Because arthritis, there are already existing database. So we are generating aging databases, aging data sets, and comparing with that. No, we do not have any of those plans. We are not looking into so much of those direction. We are more comparing, like as you ask, what is the mechanism? Why the pericytes are differentiating? And to answer that, we are trying to understand what is the signature of the aging fibroblast. But again, we are not looking at I mean, that we have not looked into.
Is the transition or the differentiation of parasites into fibroblasts, is that a lower energy intensive process? So in other words, it takes more energy to sustain parasites. And then if energy production is decreased as it is in aging, then because you can't sustain all those parasites with the amount of energy that's around, then you end up with the default state, which is fibroblasts. Like, is it an energy dependent process where you just don't have enough energy to
I'm
support?
not sure. I'm not sure. I think so. It's the support. It's the interaction between the endothelial cells and the pericytes. Because if there is a lot of senescence of the endothelial cells, that is what I think is the mechanism. There is a cell surface changes, and this is then initiating the pericyte to fibroblast differentiation. Why I'm saying that, if you co-culture pericyte and endothelial cells after certain passages, the pericytes start differentiating into the fibroblasts. So even if these kind of results can also be seen into, so for example, if you have a co-culture of pericytes and fibroblasts, it's easier to maintain them compared to if you put the pericytes alone. So there has been some reports into these directions. So I'm not sure whether I mainly think it is because of the interaction between the endothelial cells and the pericytes, and this interaction this is required to maintain their pericyte identity. When this interaction is affected, the pericyte identity of the is impacted, and then they differentiate into the fibroblast. So in terms of energy requirement, we have not looked into that.
Are there plans to look at the energetic, requirement of parasites into fibroblasts, or look at mitochondria function in the parasites versus the fibroblasts, or even I think now that you mentioned the interaction between endothelial cells and the parasites, now I wonder if uh, maybe it's either endothelial cells may be exposed to microbes because microbes can circulate freely in the blood. I mean, granted, maybe not at a threshold to impact sepsis, but and maybe that's the danger signal to the parasites, and then the parasites then differentiate. I know it's probably like a wild speculative theory, but I'd imagine that endothelial cells may send signals to the pericytes if they're exposed to microbes to differentiate into fibroblasts as a compensatory mechanism. But I don't know, are there plans to, and wait, and another, before you answer there too. So I wonder if the parasites that go senescent as a part of that process, um, cell senescence can act as an antiviral, for example, antiviral strategy. And senescent cells will release pro-inflammatory factors, basically signal to the immune system, we need help here, right? So I wonder if parasites are infected intracellular microbes, and then they go senescent, basically, especially if it's virus. I mean, you're turning off the protein synthetic machinery, but then they're senescent, right? So uh, I don't know. These may be beyond the scope of your lab, but I just wonder about these, the role of microbes
So
in
we have
this.
a lot of, we actually, we have, I mean, we don't do this, so we have not looked at, but we have like, around the LKC medicine, the labs which are, but we which look into the microbiome and so on. But we have not worked in the. I mean, we have not looked into it in these directions so far. But I mean, it's very interesting. This peripherocyte to fibroblast differentiation may be driven by this. Again, one has to probably look into it, and I think so. It's an interesting area to look into it, but we have not yet.
Nice. All right. So in going back to the energy limitation or potential, right? Because you see mitochondrial dysfunction as a part of the story. So there's emerging studies on mitochondrial transfer where you can basically,
Yeah.
yeah. So could that be part of this story, the regenerative story, where you isolate either endothelial cells, lymphatic endothelial cells, parasites, or healthy fibroblasts? And you basically, the process is pretty simple too for the mitochondrial transfer. You just centrifuge it with the cells, and some percentage of the mitochondria get into those cells. And then maybe you could re-inject that in to the animal. And if the niche and energy limitation is a part of the story for it going, senescent and aged, maybe that's a part of the regenerative story. I don't know. What do you think?
Yeah, I mean that is something to be looked into again. Also to be make sure like the animal models which we are using, because when we are re-injecting, are the pericytes properly integrating? And there are all those. Of course, there will be controls to it, but still, I mean, having the proper model where we can inject into the bone, and because injections causes injury to the bone and all those things. So I mean there would be, and then there is that triggers angiogenesis when we inject the things. So I think so that would be something to look into, where this mitochondrial transfer and so on plays role or not. People have not looked into, at least in this context.
So when you think about regenerative capacity of the vasculature, which includes the lymphatic system, what's going to be the emerging therapy that can help rejuvenate the vasculature as a means for rejuvenating bone? Like, what do you see as on the
Okay.
horizon?
Yeah, one is to be able to promote angiogenesis and lymphangiogenesis in aging. I think so that itself is sufficient. So, for example, one promote or just maintain it. I mean, the like if there is a aged individual, the issues are that it is less angiogenic, it's less lymphangiogenic. I mean, the capacity for these cells to proliferate, build on, when there is because both angiogenesis, both angiogenesis is lymphangiogenosis is required. When there is an injury, the tissue regeneration is required, and if these cells are not proliferating, they are not active, there is the problem. So if, for example, that's the difference between the young and old. The young, if there is an injury, they respond, they are proliferating, they actively go in that and they proliferate, they regenerate the tissue. And now what is here affected in both the cases, angiogenesis and lymph, their capacity to respond to the injury, their capacity to proliferate. So I think so any aspect where people can trigger angiogenesis and lymphangiogenesis, which is in a controlled way, not like, it is triggered and it's not stopping, or it's not normal angiogenesis, not lymph normal. That would be important. How uh, one can trigger angiogenesis and lymphangiogenesis that is close to normal, not something which is, for example, there are certain diseases where the lymphatic vessels grows in, they're in the bone, they degrade the bone. So I mean, it should be a normal angiogenesis and normal lymphangiogenosis, and then it has to be site specific. If there is an injury to bone, and if we promote angiogenesis and lymphangiogenosis all over the body, then also it's a problem. So I think so the current issue, the current problem is that only that if we trigger something, we are going, because everything is similar in all our organs, so we are not able to do that. We are not able to exploit what, like VEGF-A, for example, or we are not able to exploit these molecules or these growth factors because they're systemic. We don't want every blood vessel or every organ having suddenly more vessels. We only want bone to have the vessel, or in certain disease we will only want muscle, or we will only want brain to have, and at that time. But that is, we don't have that. So I think so the two things, promoting this, but most importantly, which we can promote, that's not a problem, we can give VEGF, we can promote angiogenesis, but we have to only promote in certain bones or in certain organ, and that is the problem. So when people have, like, if I have that, I can be confident, now I have a molecule which promote only bone angiogenesis, then that's it. We don't want anything. We are very sure now only I'm targeting bone, and there is increase in increase in angiogenesis, which is limited. It's not abnormal, like it's not some kind of proliferation which is abnormal proliferation, and then it's okay. We have those molecules, and it is very specific. I think so the tissue specificity is the key when we talk about angiogenesis and lymphangiogenosis. We don't have that. That's the limitation.
So easier said than done, right? How do you target it specifically to the bone and specific, the angiogenesis and lymph angiogenesis specifically to bone? How do you do that?
Yeah, so for that is what is ongoing research in the field currently, is that to understand how the vessels in different organs differ, organ specificity of the vasculature and the lymphatic vasculature. So the more we understand at the molecular level how the blood vessels in bone is different to that in any other tissue. So that's, and how in aging they differ, isn't it? So I think so understanding, which is very difficult, because there is organ specificity, but we want the molecules which are very specific only to bone. It should not be, even if it is there at certain few other organs, then also it doesn't become so specific. That means if we take that, it will be still affecting the other tissue. So I think so that is what is ongoing in the field, that understanding how the organ specificity, the tissue specificity of the blood and the lymphatic vessels at the molecular level. And more we understand how they differ from each organ, the more likely we will end up identifying the tissue specific molecules for the vasculature.
Fantastic. All right. So within that context, that we've got to wait for the, organ specificity. Are there tricks? Are there, like, what are you doing, as the expert in the field? Like, what are you doing on a daily basis? And hopefully it's more than just diet and exercise and get good sleep.
Yes.
There's got to be stuff, that you're thinking about to optimize, bone health and vascular health.
Yeah. So what we have done is, like when we have been imaging, we have identified certain molecules which we think that this is more in the bone, expressed in bone vasculature. And when we find such molecules, we generate the mice where we are, like, we have either the floxed mice or the reporter mice, where we can, so that we can do the lineage tracing. So we are doing that. For example, we have identified certain molecule where we see that it is this, we see in only two contexts, the tumor angiogenesis and the bone angiogenesis. So next stage is that we generated those mouse line to do the lineage tracing of those, like how we say. So those kinds. So basically on the daily basis, we are also, as I said, we are trying to generate some atlases, endothelial, but also pericyte and fibroblast atlases between different organs to understand how. So basically, one of the aspect is to look at the molecular level is with the single cell sequencing, that how different endothelial cells vary, or the pericyte vary, or the fibroblast, like all these associated cells varies. But then once we have this target, the next step is that, as we are doing for some of the molecules which we think are important, we are generating the reporter mice and doing the lineage tracing of those molecule with the, tagging the cells and seeing what is happening, where it is there. Because maybe we saw it in the young mouse that they are different, but maybe in the aging it's not the case. It starts being expressed in a non-specific way in other cells. So we have to look at different conditions. So the next stage is, normally when we identify such molecule, we use this preclinical mouse model to study more.
Have you done, I know you did the proteomics, but have you done the metabolomics too? And looking at,
That is the plan, because I think so metabolomics is very important, because we do see a lot of changes across the bone which are different in different bones, and bones are metabolically different also. That is for sure. We don't have any second opinion on that, but we have not yet done. We do have plans to do the metabolomics of all different bones. Yeah, there is a plan.
So wait, so when I asked about what are you specifically doing to optimize bone and angiogenesis and lymphangiogenesis and try to resist age-related change, not necessarily Anjali the scientist, like in your day-to-day, are there things that you do that maybe people would know about that they could, use in their day-to-day to try to optimize bone health in the vascular, in the vascular niche?
You mean in the research? Sorry, in research, what do you mean?
Yeah. So you've got the science that you're chasing, the mechanisms related to the vascular health and the vascular niche in bone. But then are there things that you do on a day-to-day basis that can directly impact that? Not as a scientist, but as like, and as I mentioned, you've got diet, you've got exercise, maybe it's supplements. Are there things that you are specifically doing that most people don't know about yet that could potentially improve, bone health during aging?
In a way, so you mean with the mice, or in general
No,
for
no,
myself?
in your day
Oh,
to day. Yeah.
in my day to day, for keeping
Yes.
the vascular and bone health. Okay.
Yes.
I think so. Two things are very important: having good sleep for
Hmm.
everything. I guess for everything, good sleep is very, very important. And then I think so. It's overall. I think so. It's the global health, isn't it? It's like not global. I mean, the whole health. It's also having less stress. Uh, Stress could also impact bone. Stress also affects. So I think so. Those things are very important. Sleep and keeping the stress as less as possible could have. Yeah. The, I would go for that. More sleep and being happy, less stress.
No special tricks, right? Like there's no, because I'd be thinking about, okay, if these pathways are involved in angiogenesis, lymphangiogenesis, and specific to bone, if there are molecules or if there are proteins that can be specifically upregulated, and maybe I can do it through diet.
Oh, okay, that kind of thing. No, no, I'm not doing any kind of those kind of stuff. We are not near to that. I don't think so. The diet and those kind of things where we should, of course, I mean, for bone, everyone knows we have to take, I mean, for general health, we know, like, bone calcium and all those things, milk, but in those sense, I think so. We for vascular health, I would not say any specific aspect to it,
Got it.
which
All right.
doing,
Yeah.
which I believe that it's very good, and I am following that. There is nothing
It's
like that.
it's too soon, probably, right? I mean,
Yes,
the
it's
field
too
is so
soon.
new.
It's too soon to be on that. What is the best thing? But of course, we do not want more of cholesterol.
So cholesterol bad for bone?
Yes, bad for bone, bad for vessels, for everything, isn't it?
Yeah, true.
Bad cholesterol is bad.
Well, high levels, right? You've got a whole segment of people who think higher is better, but low total cholesterol levels are low in youth, and then they peak at
Yeah,
midlife, and then they come down towards end of life. So the
yeah.
all-cause mortality data studies show that, the people at the end of life with low total cholesterol versus the people with
Mhm.
high cholesterol. So there's a whole segment of people who think high cholesterol is good because, the comorbid conditions have low cholesterol, but that's a story for another day. So
Mhm.
thanks, Anjali. This was great. I appreciate your time and I appreciate your presentation and answers. And I'm looking forward to your lab's work in the future.
Yeah. Thank you, Mike. Bye.
Have a good day.